Hello friends, this is Sonu, and today we'll discuss very important topic in the microbiology, that is the staining method or stain preparation. First of all, why we use the staining method in the microbiology for the identification of the bacteria. Without a staining, why we can't identify the bacteria? Actually, for example, that this is the slide, and these four are bacteria, just for the understanding purpose. So four are the bacteria without staining. And if this slide will keep in the microscope, light microscope, and if we will see, we can't see anything in the light microscope. Why? Because the without staining the live bacteria, we can say the live bacteria have lack of contrast. Due to the lack of contrast, the light become pass away of the slide, and we can't see anything in the slide. Due to this reason, for the identification of the any bacteria, we need a stain. That's why the staining preparation or staining method is very important to know for the identification of the bacteria or recognize the bacteria. So let's talk about the staining preparation. Staining preparation means just we can say that we color the bacteria, and due to the color, the light microscope will not pass away the light from the slide, and then we can see that something is there in the slide. Either it's a bacteria, which types of bacteria, there are structurally, morphologically, everything we can know. So first of all, the type of the stain. We have four types of stain. First, the simple stain, negative stain, impregnation stain, and then finally differential stain. Differential stain. Differential stain further divided into two parts: gram stain and acid fast stain, or other name Gill-Nielsen stain or Gill-Nielsen method. So first, let's talk about the simple stain. What is the simple stain? Simple stain means nothing, because in the simple stain, let's talk about the simple stain. In the simple stain means, this is the slide. Let's before the start the stain, some basic thing we have to know. Before staining, we take the smear. We prepare the smear, or we can say we take the specimen. What is the smear? Smear. Actually, for the identification in the microscope, by the microscopically identification, we need slide. So we take slide first. We take slide. Second one, wash the slide by soap or maybe detergent. After the wash, third, dry the slide, completely dry. Dry the slide by fire. Means in the burn, in the, we can say, like this is the, for example, this is the beaker, and by this beaker we can put in the slide like 1 minute or maybe 30 seconds or 50 seconds, and then we can completely dry. After then we can start to the staining method. So this three group, or we can say this three step is the smear. This is called the smear. So always whenever we in a microbiology, whenever we're going to culture, or we're going to start the staining method, or we're going to identify the bacteria, first we take smear. Before the putting the bacteria in the slide, this three steps we have to done. We should done. Like first we take slide, we completely wash by the detergent or by the soap. After that, by completely dry. After the dry, then we can put the bacteria, and then further we'll do the step. So let's now with the simple stain. What is the simple stain? Simple stain means nothing, just we take smear, means we take slide, and in the slide, by the crystal violet. Crystal violet. What is the crystal violet? Crystal violet, that is the blue color, or gentle violet. Gentle violet, or we can say methylene blue. Methylene blue. These three molecules we take a slide and we take the bacteria for the identification that this bacteria is the gram negative, gram positive, or other bacteria. So we take the bacteria here. For example, these are 4 bacteria, and then by this 3, anyone like either crystal violet, gentle violet, or methylene blue. By the dropper, we put in the slide. For example, like this, like that, we put it. After the put it, for 1 minute or 2 minutes, so maybe maximum 60 seconds. After the 60 seconds, this slide washed by water. Washed by water. Then we can see in the light microscope. But one bad thing, then we can identify that something is there. Maybe either it's a bacteria or maybe other microorganism. But we can't identify that this is the gram negative or this is the gram positive, because the gram negative bacteria is the red color, or gram positive bacteria that is the violet or purple color or blue color. So just blue color in a slide we can say, nothing else, just blue color. That's why this stain method is not successful. Then we further move in the negative stain, because by the simple stain we didn't successfully stain that this the bacteria is the negative or positive. Just we can think that this is the bacteria, but which bacteria we don't know. So let's move in a different stain. Different stain, the negative stain. Negative stain. Again we prepare the same here, and in this stain, the same molecule, crystal violet, gentle violet, and methylene blue, the 3 of 1, when again apply in the slide. But actually these 3 will not color the bacteria, will color the background of the slide by this staining method. The negative staining method will not color the bacteria, will color only the background of the we can say slide. So it's look like that. This is the background color. And for example, this 2 is a bacteria. So now in a microscope, again the same problem. In the microscope we can see that the all except the bacteria, all background is the blue and the bacteria is the white. So again the suspense that this bacteria is the gram positive, gram negative, or which types of bacteria. So again this method is not success. Then we'll move another method, that is the impregnation stain. Impregnation stain, let's see that this staining method. In this staining method, not use any crystal blue and methylene or other gentle violet. In this stain, just take the slide and use silver color. Use silver color. What is the role of the silver color? Actually, whenever put the silver color in the slide, so the that area, that bacteria has become thick. So by the help of the silver color, we can say bacteria is thick. So by this method we can identify or we can determine the flagella of the bacteria. But this also not successful. For example, in a spirochete species, for example, just take a Trypanosoma pladome, that is causative agent to syphilis. So Trypanosoma pladome, if we take the bacteria in the slide and that bacteria, the Trypanosoma pladome, then or we put the silver color, so we can demonstrate or we can determine that this bacteria is the Trypanosoma pladome. But just exception, if another bacteria will be, so we can't identify. So we can say by this method somehow we can identify flagella of the bacteria. So flagella of bacteria, any bacteria who has flagella, then we can identify. But this method also is not, we can say, so widely used or so best method for the staining preparation for the identification of the bacteria. That's why this method also nowadays not used very frequently. Now last one, that is the differential stain. This staining method is the very popular and very good method for the identification of the bacteria. That's why nowadays we use these types of stain. As you see that the differential stain is divided into two groups, gram stain and acid fast stain or Gill-Nielsen stain. So this acid fast stain or Gill-Nielsen stain, actually this stain only for the MTB, or we can say mycobacteria. Only exception this method, mycobacteria. For this species we use this method because by the gram staining we can't identify this types of bacteria. So you can obviously you can think that mycobacteria is the Mycobacterium tuberculosis, Mycobacterium leprae, Mycobacterium bovis and other Mycobacterium species. So by this staining method we can identify this types of bacteria, and rest of this bacteria, all gram positive and gram negative bacteria can be identified by the gram stain. So today we'll just focus on the gram stain because this is the very huge staining method and used in all over world. That's why let's talk about the gram staining. So we should rub this one. First we take smear, or we can say we take a specimen. After the specimen means this is the slide, and for the understanding purpose just we take two bacteria. So we put bacteria in the slide, then by the heat fixation, so we fix so just example this is the candle, and we can say heat fixation, because by the heat fixation bacteria will fix. It will be 60 seconds or maybe less. After the heat fixation, second step, we mix crystal violet. Crystal violet. We mix crystal violet. Means by the dropper, we put on the dropper in the slide and just rub down, or we can say just like that we can do, and the all liquid will spread on the all slide. After the crystal violet, mixing of the crystal violet mix, we should write crystal violet or methylene blue or gentle violet, out of three anyone can be used. Then after the crystal violet, wash by clean water. Clean water. So we should keep the crystal violet 60 seconds. After the 60 seconds, wash by the clean water, and then next step, add iodine. Iodine for 60 seconds. Iodine for the 60 seconds. After the 60 seconds, again we'll wash. Wash by water. That is the clean water. After the wash, then again we'll put alcohol. Alcohol 95% of ethyl. Ethyl alcohol. Again for the 60 seconds. After the add the alcohol. After the 60 seconds, again we'll wash by water. After the washing water, we put the safranin. Safranin. This is the red molecule. This color is the red. And after the 60 seconds, again the timing the same. 60 seconds. We take out from the safranin. This is the slide. And as I said, there's two bacteria. One and the another one is the red, because we will discuss that how it's red and how it's blue. So red is the gram negative bacteria and blue is the gram positive bacteria. It's look like blue or maybe purple or violet. So this is the gram positive bacteria. Now we can see in the microscope, light microscope. So we can see that this is the red, means the gram negative bacteria. This is the blue is the gram positive bacteria. Or if the curve shape, so we can say this is different types of bacilli bacteria. The cluster shape, the staphylococci bacteria. Like that, by the shape we can identify. Now we'll see that how by this all procedure, how it's a color, the red color, and how the bacteria will, the gram positive bacteria will be the blue color. So let's discuss. Let's rub this one. First, we should know that gram negative is red color. Then gram positive is blue color or violet color or purple color. So during the this procedure, how the color change. So let's talk about first. We should make bacteria here. So this is the bacteria. As you can understand, think about that this is the peptidoglycan layer, and the linkage between the peptidoglycan, the transpeptidase enzyme. So this is the transpeptidase. This is the gram positive bacteria. And let's talk about here. Again, we'll make bacteria. That is the gram negative bacteria. It's a very less peptidoglycan layer, not very high like gram positive. And the additional layer, that is the lipid layer, additional cell wall, which made by the lipopolysaccharide lipidase. That's very toxic. So this additional layer is not present in the gram positive bacteria. Now let's start. For example, one is the gram positive and one is the gram negative bacteria. Gram positive and this is the gram negative bacteria. Right now we don't know which is the gram positive and gram negative. Just we know the two bacteria. So first we take the bacteria and then we'll mix the crystal violet. So whenever crystal violet will mix, so this is the bacteria structure. So crystal violet will add in the peptidoglycan layer, and by the small porine, crystal violet can also will enter in the gram negative bacteria and will make bond like structure in the gram negative bacteria. But in a gram negative bacteria, the crystal violet will go inside and it will live in the very far to each other. So we can say the distance between this crystal violet and this crystal violet is very far as compared to gram negative, gram positive. In a gram positive bacteria is the very near bonding of the crystal, means the very near to each other, but in the gram negative bacteria is very far. After the crystal violet, after the wash, and again we add the iodine. What will do the iodine? Let's see. So the red one is the iodine. So iodine and crystal violet will make complex, tightly complex, which is, you can see that is a very tightly complex, or we can say will bond like complex made in the peptidoglycan layer in the gram positive and as well as in a gram negative. But in a gram negative it's a very few, because these gram negative bacteria don't have peptidoglycan layer, very thin peptidoglycan layer. But in a gram positive bacteria, a lot of peptidoglycan layer. And as we know that the crystal violet and the iodine will make complex in the peptidoglycan layer. Due to this reason, here is the very less and here is the very high. After the iodine, Again we'll wash, and after the wash again we'll add the alcohol. Actually, what did the alcohol? From there we can identify the gram positive and gram negative bacteria. So alcohol, as we know that alcohol is the lipid soluble molecule, and this in a gram negative bacteria, the outer layer, which is the lipopolysaccharide, which also made by the lipid bilayer, that's why alcohol can break down this lipid bilayer. But in the gram positive bacteria, no any problem, because the gram positive bacteria don't have any cell wall, so don't have any lipid layer. That's why the alcohol will not affect on the gram negative, I mean positive bacteria, but this will affect on the gram negative bacteria. So let's discuss about alcohol. Actually, alcohol will, it is the lipid soluble, so alcohol will be break the this cell wall. So whenever alcohol will come, this cell wall now break down by the help of the alcohol. So alcohol is the break down the outer layer wall, and actually after the break the outer layer wall, that is the lipid layer, now the alcohol will also destroy the complex of the iodine and the crystal violets. So whenever it will destroy the complex, the bacteria will discolor, means don't have any color. So let's, now the alcohol will do that, now they don't have any crystal violet, bacteria is the now discolor without any color, just in normal condition. So in this time, means during the washing, the gram positive bacteria is still in a blue, because the alcohol will not do anything here, because the very tight bonding, so it will not affect in the iodine complex. But in the gram negative bacteria is very thin peptidoglycan layer, and due to the disruption of the outer layer wall, they will also break down the complex of the iodine and crystal violet. That's why in this situation, if you see the microscopic here, just you will see the 2 gram positive bacteria. You will not see in a gram negative bacteria, because due to the decolorization after the alcohol, putting the alcohol after that. So now here gram positive bacteria already we identified that this is the gram positive bacteria. But for the gram negative bacteria, again we'll add safranin or red color, because the safranin is the red color. So safranin obviously will go here, and it will be the now colorized by the red color, because it's already blue, so red will not affect in the blue area. This will affect on only the gram negative bacteria which already decolorized by the alcohol. So that's why this slide will put in the safranin, or maybe saffron liquid by the dropper, it will put on the slide and spread on the slide. And after the again wash, and after the washing, now we can see in the slide that this is the gram negative bacteria, which color is the red color, and this is the gram positive bacteria, which is the blue color. So by this method we can identify that which bacteria is the gram negative, which is look like the red color, that is the gram negative bacteria, and which look like the blue color or violet color or purple color, this 3 is the gram positive bacteria, which belong to the gram positive bacteria. Even we can see in the light microscope by the shape, by the size, so we can more identify that this bacteria is the cocci shape, bacillus shape, curve shape, and different shape. So these are the main important gram staining method that we have to know. Now we'll discuss about some of the bacteria which is not even stained by the gram staining method, which is not useful for the gram stain. That is the exception. So first of all, let's talk about those bacteria who is nasty bacteria. That is the spirochete. Spirochete. In a spirochete, Trypanosoma pallidum. Trypanosoma pallidum. Trypanosoma pallidum. This bacteria is the spirochete, means look like that, spiral. So whenever you will do the gram staining, it will not stain very well like the other gram positive and gram negative bacteria. That, and after the staining method, if you will see the light microscope, it will light become pass away and we can't identify. That's why in this condition we use dark microscope. Dark microscope. In the dark microscope, we can identify that the spiral shape bacteria are there in the slide, so we can identify that this is the Trypanoplasma. Or by the dark microscope or fluorescent antibody. Fluorescent antibody. These two methods can be identified. Actually, whenever we will discuss about the Trypanoplasma, on the time we will very details will discuss the how we can identify, because a lot of method can be identified the Trypanoplasma, even all bacteria. So on the time, obviously we will discuss everything in details, but just you can understand that by the dark microscope or fluorescent antibodies can be identify the Trypanoplasma. Then next bacteria, that is the Rickettsia. Rickettsia. Actually, Rickettsia is an intracellular organism. It's live in the inside our cell. That's why by the gram staining we can't identify, because the gram staining just identify the outer layer. So this is live in the inside the area. So that Rickettsia. Next one, Chlamydia. Chlamydia species. The Chlamydia species also is not well we can say well identified by the gram staining method. After that, Legionella. Actually, Legionella can be identified the gram staining method, but in that time we have to increase the time of the, like, during the crystal violet normal time is 60 minutes, during the Safranin V8 60 minutes, but in this case we have to increase the time, maybe 2 minutes, 5 minutes. On that time, then we can identify the Legionella species. So these are the exception bacteria which are not the normal method of the gram staining. By this method we cannot identify these types of bacteria. So these all information about the gram staining method that we have to know, we should know. And in next part we will discuss about the acid fast stain or Gill Nelson staining. So thank you so much.